Okay, in this segment, let's actually talk about the types of variation that we see in the genome. In terms of counting, the most common variation we see is something that we call a single nucleotide polymorphism, ubiquitously known as a SNP, an SNP. Some people talk about single nucleotide variation, SNV. I'm not even going to write it on the board. SNP is the word that people use. This is a single letter in the genome that has changed, that has more than one possible allele. So you might have the genome with a T, but other people at the same locus, the same position on a chromosome at a certain position, have a different letter. This would be called a TG SNP. And most SNPs are biallelic, two choices, T or G. In other discussions, I've talked about the rare triallelic SNP, but it's very rare. Most SNPs are biallelic. Now, we call them polymorphisms because they are not new and they are not rare. So what do I mean by that? We might look at a population of humans. We might sequence or test their DNA at this position, and we might find that 90% of people have a T and 10% of people have a G. Therefore, we would call G the minor allele because it's the smaller in frequency, and we could call T the major allele for that population. It is possible that another population would have 80% G, and therefore G would be the major allele. So whenever you're talking about major and minor alleles, you must specify the population upon which these were measured. Okay? So how common is this? I told you it was the most common. If I take two random humans on Earth, sequence their genomes, and compare the two genomes, I will see that they will differ at 4 to 5 million locations in their genome by a single base. So a T versus a G. Now let's be a little bit more precise. Everybody is going to be diploid. So when I tell you that there's two choices for T and G at this position, that means that when I sequence you, I might find that you're a TT, which means you got a T from both mom and dad. I might find that you're a TG or a GT. You don't know which one you got from mom and which one you got from dad, but those wind up looking the same because we can't distinguish. Or you might be GG. So there are three, these are called genotypes. And there are three possible genotypes for a biallelic SNP. So when I'm saying that you take two people and compare them, 98% of their genome, 99% as I said, will be identical. At the 1% that could be different, there will be about 4 to 5 million locations where they don't precisely match in their genotype. That means you are a TT and the other person is a TG, or you're a GG and the other person is a TT, etc. So that's a good number to remember. You'll have about 4 to 5 million SNPs that will differentiate you from any other human. Now, I should say that if you're comparing yourself to a sibling, those numbers get much smaller because there the differences are just based on what you inherited differently from your two parents. But I'm talking for unrelated individuals. So that's a SNP, most common variation in the human genome. It's also the most easily measured. And I must say there's a certain looking where the light is shining. Because we can measure SNPs so well, we tend to focus on SNPs in a lot of our scientific studies. That's fine, but we should acknowledge that part of the reason we're focusing on SNPs is we're good at measuring them. There are other things that I'm about to discuss, more difficult to measure, not necessarily less important. So the next one to talk about would be an indel, which is a short form of insertion deletion. The kind of fun thing about indels is they're called indels because you don't really know when you look at two people if one person has an insertion or the other person has a deletion. Let me show you what I mean. An indel might look like this. You have identical sequence in the two humans, but then somebody has AGCTTA, and then it goes on. And then this person might have nothing, dash, 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 which means really this connects directly to this. So they're missing that. So it's called an indel because from this person's perspective, that's an insertion that the other person has that I don't have. But from this person's perspective, hey, what happened to you? You got this deleted. So unless you go back and understand the historical things that happen to lead to this, you can't tell if it's an insertion or deletion, so you call it an indel. These occur in the human genome. As you might expect, there is a spectrum of short indels all the way to very long indels. There are deletions that can be hundreds, thousands, even tens of thousands of bases. The most common would be 1, 2, 3, 4 bases. And our ability to measure them is okay, but it's not great. But they definitely occur in the population, and they can obviously definitely change a protein. If they're sitting in the middle of a protein, that's roughly 2 amino acids worth of DNA that you've just cut out. Or even if it's occurring in the non-coding region, it can affect things like promoters and other protein recognition sites that are involved in turning genes on or off. You can have entire gene deletions. Really, it's just a type of indel where the whole gene, so this is how I've been drawing genes, the whole gene gets deleted. So instead, you have A and B, and now you have A and B right next to each other, and the gene is missing. And it is startling how many people are walking around perfectly healthy, missing entire genes. Obviously, those genes are not key for life. There are genes that, if they're deleted, are not compatible with life, but many are not necessary. The opposite of that, in some sense, is copy number variation. Very important for pharmacogenetics. There are cases of genes where some people have the one copy of the gene, but other people have 2 or even 3 perfectly functional copies of the same gene. Okay? That is interesting because, if you will, the dose of this gene activity, you get 1 dose here. This is twice the dose, so you almost have twice the amount of that gene in terms of whatever it does. And this one might be 13. Just to give you an example, there's a famous cytochrome 2D6, which is one of the metabolizing enzymes in the liver. It's a member of the cytochrome family that we have discussed in other segments. Cytochrome 2D6 is involved in the metabolism of hundreds of drugs that it metabolizes. Interestingly, there is somebody who has been reported in the literature who has 13 functional copies of this gene, CYP2D6. So that person has 13 times more ability to metabolize these drugs than somebody who has only 1 copy. And that has huge repercussions for their drug response for all of these hundreds of drugs. And in fact, they were discovered because they were taking a cholesterol drug, and they needed 10 times the normal dose to get the benefit of that drug. It was simvastatin. And you can Google or do a PubMed search for simvastatin and copy number variations on CYP2D6. The other thing I'd like to tell you about are structural variants. Structural variants are large-scale changes in the chromosome. So I'll just say large-scale change in chromosome. So, for example, I might have a chromosome that I'm dividing into section A, B, and C. And in some people, that same chromosome might have A, but then the B segment is in reverse order. So I'm going to just draw the B backwards. And then C. So this one goes arrow, arrow, arrow. I should do that in a different color. Arrow, arrow, arrow. And here it's arrow, arrow in a different direction, arrow. So this can be hundreds of thousands or millions of bases. Now, all of that DNA in the middle is still perfectly good and can be used to make genes and stuff. But at this junction and this junction, things look different because I've changed exactly what's happening at that boundary. That could introduce mutations that are either neutral and nobody cares, or are in key genes that have now been disrupted and can have big consequences, for example, for cancer risk or other things. And so that is one kind of structure. That's just an example of a structural variant. Another example would be you just change the order. So now instead of A, B, C, you have A, C, B. And again, this can happen on a very large scale, hundreds of thousands of bases. And again, everything in the middle of B and C might be fine, but this junction and this junction have been changed and may have introduced changes in proteins that could have an effect on health. Another and the final kind of very important for disease variation, which is a kind of insertion, is a triplet repeat. So it turns out that one of the things you can see is a position in the genome where some people have CAG, for example, and other people have CAG, CAG. So that's a double repeat. And other people might have 6 copies. And I'll just write it as CAG to the 6th, meaning CAG, CAG, CAG repeated 6 times. So each of these is the allele. And for some diseases, the length of the repeat can determine whether or not you get disease or not. The most famous being Huntington's disease, where there's a triplet repeat. I don't remember the 3 letters, but if it gets longer than a certain length, then you have Huntington's disease. If it's less than that, you don't have Huntington's disease. And in fact, the severity of the disease is related to the length of the triplet repeat. So this is not absolutely exhaustive, but it gives you a sense for all the ways in which human genomes can vary. And these are all variations that could impact PK genes or PD genes, and therefore could impact drug response. And that's why they're worth considering. The final thing I want to mention is this idea of haplotypes. And we'll do that here. So I keep talking about mom and dad and how they gave you some SNPs, and maybe they gave you some structural variations, and they gave you some gene deletions. And let's just think about a SNP because it's easier. And let's imagine that we measure your DNA at 3 positions. And we see that you have a GT SNP at position 1. This is position 1. At position 2, you have an AG SNP. Mom and dad, one gave you an A, one gave you a G. Here, one of them gave you a G, one of them gave you a T. And then a SNP that's a TA SNP. Everything else is identical in this little loci of DNA. This is interesting and good to know, but it is not sufficient to tell which of the SNPs came from which of the parents. So for example, mom may have given you GAT on her chromosome, and therefore dad must have been the TGA, mom, dad. It's not clear here, so I'm going to put question marks. That's one possibility, possibility number 1. But as you now can see, there are several possibilities. Another possibility that would yield the exact same genotype, this is called genotype, is mom could have given you GGA, And dad could have given you TAT. And you can spend quite a bit of time generating all the possible combinations that would lead to you having a G and a T at position 1, an A and a G at position 2, and a T and an A at position 3. The detailed identification of which alleles occur on the same genome or chromosome, so these are on chromosomes, and one chromosome is from mom, one is from dad, is haplotype. So just to define haplotype, it's the alleles that were together, were or were together on same parental chromosome. So in general, you're getting a chromosome from mom and a chromosome for dad. And for any allele, you'd like to know, did this come from mom or for dad? You only know that information if you have the haplotypes, because that tells you the alleles that are together on the same parental chromosome. Most of our genotyping technology will tell us the 2 alleles that you got, but will not put them in phase. So if you know the haplotype, it's called phased. And I don't know why they use that word, but what it means is you know all the ones that were on mom and all the ones that were at dad. This can be important, and let me just end by telling you how this could be important. Let's say that G messes up a protein and the A messes up a protein. In this scenario, mom has messed up her protein twice, once here, once there. So mom's giving you a messed up protein that doesn't work, but dad has given you a fine protein because he doesn't have any of the mutations or SNPs that mess up the function. Okay? In this case, mom has one variation that messes up the protein, and dad has the other. So you're going to have 2 copies that are messed up, and you will not have a functioning protein at all. Here you have half the dose. Here you have 0. So haplotyping can be very important when looking at deleterious, that is to say mutations that destroy protein function, because knowing whether they're on the same or different chromosomes allows you to see if you just have redundant hits on the same protein versus independent hits on both mom and dad's copy. And obviously in the context of genetic disease and also in pharmacogenomics, knowing the haplotype phasing can tell you if you have a little bit of a problem or a big problem.